All right, I think we should get started. We already have quite a number of audience joined. So hello everybody, welcome to our seminar today. My name is Yare. It's my first time hosting this journal club. I'm the student volunteer leader in the eastern coast of Dashu. So it's been a great privilege for us to have invited Dr. T here today from Hopkins. So before we get into the presentation, let me give you a very quick overview introduction of Dashu. So Dashu is a 501c3 nonprofit organization, and so that we have the tax exempt status by federal and by California state. We are a very young organization that is founded 4 years ago, and Dashu is dedicated in promoting data science education, providing a virtual community for people who are interested in data science, and build a platform for people to make social and developmental connections. And we also have many strategic partners, including American Statistical Associations, BASE and CAPS, and etc. The good news for everybody here today is that our membership is free, so long as if we have the pleasure of having you attending any of our events, you will be automatically enrolled as an active member. And if you're interested in funding us or supporting us, we can make sure that our administrative cost is very low. We can make sure it's below that 1%. All of the work, all of us, we work as volunteers. So if you're interested in funding us, supporting us, all of the funding will go to serving the community. And yeah, I can see you're sharing the slide. So we organize a lots of different kinds of events. We sponsor and host many international scientific conferences. And monthly, we're trying to do this virtual journal club. We have amazing speakers invited from industry, from academia to give talks. And we also hold several technical seminars and career development events. So for many of you who are not very familiar with how we propagate the specific event, you should definitely check out the website, check out, you can also email us for any questions and suggestions. And if you use WeChat, please subscribe to our official account. You see a QR code in here. Now put up your phone and scan the QR code. So you will be notified whenever we have the first-hand event as a notification. About the journal club that we're hosting today. We have speakers and topics, great speakers from academia industry, as I just mentioned, and we still need a lot of your support to help promoting the events. So whenever you see something that is posted by us from social media, please subscribe it, please share with your friends, and we also are looking for sponsors that are welcome. If you're interested in our past events, we always put up the recordings and slide deck that everybody, whoever is a member, have the free permission to download them from the dashu.organization. And also, please subscribe to our WeChat official account, and you can see what are the past events and what are the events that are coming up. So before I get down to introducing Dr. D, I would love to remind everybody that of the upcoming event later this May. That is, it's not virtual anymore, it's in reality. For many of you, if you happen to live near the Bay Area, you should definitely check out this event that Dashu and the ASA San Francisco chapter is co-sponsoring this event that is focusing on the real-world significance beyond p-value, following a very important paper that was just published on Nature discussing should we abandon, or is it the time that we abandon this p less than 0.05 criteria. And we have a lot of amazing speakers. Here's only listed a few. That we have speakers from ASA, from Stanford, from Genentech, and they're going to be focusing on this very statistical kind of question. So for many of you who happen to live nearby, definitely check out this event. You can register one day before the event, May 21st. That being said, put this onto your calendar. If you have time, definitely check out. All right, last but not the least, let me introduce today's speaker, Dr. Hongkai Ji from Johns Hopkins University. He's going to give this, I believe is going to be an amazing talk on global prediction of gene regulatory landscapes using multiple sequencing platforms. So a quick overview of Dr. T's resume. I'm just looking at a fantastic CV in here. Dr. T got his undergrad in engineering automation from Tsinghua University, and he received his PhD in statistics from Harvard. About 10 years ago, he joined as faculty at Johns Hopkins University and started to serve as director of the biostat department since last year. And today's topic is focusing on how to integrate multiple platforms from sequencing experiments to generate or prediction or infer biological pathways using multiple platforms. Personally speaking, I work a lot on bulk sequencing, and our lab works on a lot of single-cell sequencing. I know single-cell technology is very new. The data analysis is already very difficult, not to mention how can we integrate that into the bulk seq. So I am very excited and have high expectation for this talk. I believe many of the audience do. So I will now ask Dr. G to give this fantastic talk. So Dr. G, I'm gonna stop sharing my slide, and if you please, you can start sharing your slide.
Okay.
Thanks.
Yeah. Let me start my screen sharing. Can you see my screen now?
Sorry, I just muted myself. Yes, we could see your screen. You can start playing it.
Okay. First, I want to thank Dashu for organizing this event and also for inviting me to give this presentation. And today, I'm going to talk about our work on predicting gene regulatory landscape using gene expression data. And first, some background. Why do we want to do this? And we know that we are interested in studying how genes activities are controlled. And as you probably all know, that in our human genome, there are about 20,000 to 25,000 protein coding genes. And the activities of these genes are tightly controlled both temporally and spatially. And at each particular time point during the development or in a particular tissue or organ, only a subset of genes are activated. And which subset of genes are activated and at what level they should be activated are both tightly regulated. And an important goal of functional genomics is to understand the programs that controls these gene activities, because this understanding will help us understand the human diseases. And the gene actually can be controlled at multiple levels, and the critical step to control gene regulation is the transcriptional regulation. And what is transcriptional regulation? Transcriptional regulation is a specific type of regulation mediated by transcription factors and cis regulatory elements. Transcription factors. So can you see my mouse? Because I don't have other things to point. So if you can see my mouse, you can see like transcription factors are special class of proteins that can bind to DNA sequences. And when they bind to DNA, they can either turn on or turn off the expression of nearby genes. And in the human genome, there are about 1,400 or 1,500 transcription factors. And each transcription factor can bind to thousands to tens of thousands of different places in the genome to control thousands of different genes. Each transcription factor can control 100 to 1,000 of genes, and different transcription factors can bind to different places in the genome and therefore control different sets of genes. And even for the same transcription factor at different time point or in different tissue or organs, they can control different sets of genes. As you can see, this is a very complex regulatory program. And in order to fully understand transcriptional regulation, therefore, it is very important to map genome-wide transcription factor binding sites for all transcription factors and in all possible biological contexts. Here, biological contexts are defined as combinations of time, space, and treatment conditions, environmental stimuli, and so on and so forth. And you can see that this is not an easy task. Currently, biological technologies, if you just do use current biological technologies to map transcription factor binding sites, you cannot actually get the comprehensive map. And the state-of-art technology for mapping genome-wide transcription factor binding sites is a technology called ChIP-seq, or chromatin immunoprecipitation, followed by sequencing. And what this technology does is, for one transcription factor and one sample, it tells you the locations where this transcription factor bind in the genome. And although it is high throughput in the sense that it can map genome-wide binding sites of one transcription factor, this technology does have a lot of limitations. And the first limitation is that it can only study one transcription factor at a time. And in each experiment, you only analyze one transcription factor in one sample. And usually that means you can only study one cell type or one condition in that experiment. And it also requires high quality antibodies, which are not always available. And also the conventional ChIP-seq technology require a large amount of cells and a large amount of input material, usually like millions of cells. And when you study precious clinical samples or like early embryos where the number of cells are small, this technology sometimes do not work that well. So because of all these limitations, currently, if you want to use ChIP-seq to study all transcription factors in all possible biological contexts, it's just very difficult. And recently, there's another class of technology that were developed for studying genome-wide gene regulation. And this technology tries to assay chromatin accessibility. And the rationale behind this is because we know active transcription factors often are associated with open chromatin. And therefore, if you see that a genomic locus has open chromatin, you can sort of infer that maybe there is a protein-DNA interaction, or in other words, there might be a transcription factor binding event. And currently, people have developed a number of technologies to survey chromatin accessibility in a genome-wide fashion. And two most widely used technologies are DNA-seq and ATAC-seq. And without delving into detail of what this technology does, you can see that here, these are the data generated by these technologies. Essentially, it's pretty much like the ChIP-seq data. And whenever you have a peak here, you know that there is a protein-DNA binding event here. And the difference between these two technology and the ChIP-seq is that it surveys the binding sites of all proteins at a time. So it's not limited to a specific transcription factor. And therefore, using this technology, you can assay genome-wide binding sites of all transcription factors in one experimental assay. Okay. And a limitation of this technology is that when you see there's a peak here, you don't know exactly which transcription factor is sitting there. So you don't know the identity of the transcription factor. However, we know that for many transcription factors, there are specific sequence motifs recognized by them. So these are called transcription factor binding motifs. And computationally, people actually can map these DNA sequence motifs or DNA patterns into the human genome. And by combining the DNA motif information and the DNA-seq or ATAC-seq information, actually we can infer which transcription factors are binding to which location. And in this way, by combining the experimental DNA-seq or ATAC-seq assay with computational motif analysis, people may infer genome-wide binding sites of hundreds of transcription factors simultaneously in one experimental assay. So obviously, this offers more information. However, we still cannot answer the question we want to answer. That is, how can we know that the genome-wide binding sites of all transcription factors in all biological contexts? That's because both ChIP-seq technology and DNase and ATAC-seq are still low throughput in terms of analyzing samples. So each experiment can only analyze one cell type or one biological condition. They can not analyze tens of thousands of different biological contexts which are combinations of developmental time and tissue and organ and other like experimental conditions. And in order to survey all these biological contexts, you need to do this experiment many, many times. Obviously, no single lab will have the resource to do those experiments. That's very expensive. That's very time consuming, and it requires a lot of expert and human power. So it's simply not possible right now. And then this leaves the question, how are we going to study this comprehensive map, the gene regulatory landscape in the human and in other species? So we have thought about this. We think that although current genomic technology may not allow you to do this, maybe as a data scientist, we can have our own unique contribution. So we propose that we might be able to do something here using prediction. So our approach is we are going to predict using gene expression. So our goal is to predict genome-wide cis-regulatory element activities or transcription factor binding sites using gene expression. So you may wonder, why do we want to use gene expression? And there are multiple reasons that we choose gene expression as a predictor. First, gene expression is the most widely measured functional genomic data type. When you look at the public database like a gene expression omnibus or sequence read archive, you can realize that the number of gene expression samples stored in these public databases are orders of magnitude higher than the number of experimental regulome samples such as ATAC-seq, DNA-seq, or ChIP-seq samples. And technology for measuring gene expression is relatively mature. And also gene expression, later we'll talk about it, gene expression can also be measured with relatively high quality in samples with a small number of cells. And these days, people also routinely do single-cell RNA-seq, which measures gene expression in single cells. That means if you can use gene expression to predict gene regulatory landscape, you can immediately expand the number of biological contexts for which you will have the gene regulation information. Okay? So that's why we want to use gene expression to do the prediction. And so this slide shows our overall strategy, how we are going to do it. We know that projects such as ENCODE and the Epigenome Roadmap, they have collected a large amount of functional genomic data for many different cell types and experimental conditions. And for example, in ENCODE, we have a number of cell types for which people have collected both DNA-seq data and the gene expression data. Here, I'm showing exon array first, because that's our first study, and then later I'll talk about RNA-seq. So we hypothesized that maybe we can use this data collected by projects such as ENCODE to train a prediction model such that we can use gene expression to predict genome-wide DNA-seq profiles. And if we can train such a prediction model, maybe then we can apply the trained model to gene expression samples stored in public databases so that we can immediately predict genome-wide regulatory landscape for many different samples, and for which people don't previously have the DNA-seq or ATAC-seq or ChIP-seq data. And then after you predict the chromatin accessibility, you can then combine that information with the DNA motif information to predict genome-wide binding sites for many different transcription factors in many different biological contexts. So that's our overall strategy. Does it work or not? So we actually first started with a pilot study where we tried to use exon array data to predict DNA-seq. So in this pilot study, what we did is we collected 57 different ENCODE cell types for which people have generated both DNA-seq data, which is called DHS here, and gene expression data. And here gene expression is measured using exon array. So after some pre-processing, we have organized the data into two matrices. And here, this matrix, the green matrix, is the gene expression matrix, and the orange matrix is the DNA-seq matrix. And each column represents a cell type, and we have 57 different cell types. And in the gene expression matrix, each row represents a gene. So the matrix basically tells you the gene expression levels for each gene in each cell type. And in the DNA-seq matrix, the yellow matrix here, each row represents a cis-regulatory element. And the matrix tells you the activity of each cis-regulatory element in each cell type. Here, activity is measured using RNA-seq. In other words, it is the open chromatin measure associated with that cis-regulatory element in that cell type. Our goal is to use this green matrix, the gene expression matrix, to somehow predict this orange matrix, the RNA-seq matrix. Okay, and essentially what we want to do is for each genomic locus or for each cis-regulatory element, we want to construct a model such that RNA-seq hypersensitivity or open chromatin can be represented as a function of gene expression. So the simplest way to do it is to use a neighboring gene approach. In the neighboring gene approach, if you want to predict the activity of one cis-regulatory element, what you do is you first identify its neighboring genes, okay? And then you extract the expression levels of those neighboring genes, and then use the training data you have. You can then train a model, for example, a regression model that help you to convert the gene expression values to the predicted activity levels of a cis-regulatory element. And we call this neighboring gene approach. Neighboring gene approach is a very natural approach because we know that cis-regulatory elements often controls the expression of its nearby genes. However, later you will see that this approach is not the optimal approach because after some exploration, we found that not all information useful for prediction is contained in the neighboring genes of a cis-regulatory element. And for that reason, we explored another approach where we use the whole transcriptome information. That means we use the gene expression levels of all genes as predictors to predict the activity of each cis-regulatory element. So that's a so-called a whole genome approach or genome-wide approach. So if we want to use all genes as predictors, we are going to face with a challenging high-dimensional problem. So essentially, we want to use a gene expression vector to predict a chromatin accessibility vector, and both vectors are high-dimensional. In the human genome, we have tens of thousands of genes. Therefore, the gene expression vector is of dimension of 10 to the 4th. Okay, and for the chromatin accessibility, we want to predict the activity of each cis-regulatory elements. There are millions of regulatory elements in the human genome. Therefore, the outcome vector Y is also a high-dimensional vector with dimension in the order of millions. And the training sample size actually is very small compared to the dimension we have here. And in the ENCODE, there are about a couple hundred of cell types with both data types. And when we did this pilot study, the training sample size is less than 100. Actually, as I mentioned, we only have 57 training samples at that time. So you can see this is a very challenging ultra high dimensional problem where you have small sample size, but you have high dimensional predictors as well as high dimensional outcome. So how are we going to handle this problem so that we can both create accurate predictions and create a computationally efficient algorithm? We actually tried many different methods, including many off-the-shelf methods such as random forest and lasso and other things. And eventually, after we explore many methods, we found that this approach shown in this slide actually works best. So we call this approach BIRD, or big data regression for predicting DNase1 hypersensitivity. In this approach, what we did is we first clustered the correlated genes into clusters, or in other words, we group co-expressed genes into clusters. And for each cluster, we use the mean gene expression value of all genes within that cluster to summarize the cluster level activity. So in this way, we essentially reduced the dimension of our predictors. So originally we have tens of thousands genes. If you cluster them into 1,000 or 2,000 clusters, you reduce the dimension to 1,000 or 2,000. Okay. So why do we want to do clustering? Clustering actually has multiple purposes here. First, obviously, it reduces dimension. And second, we know that genomic measurements are very noisy, and especially at the single gene, individual gene level. If you do clustering and average gene expression levels within each cluster, you average out noise. So reduce the noise level, or in other words, you increase the signal to noise ratio for the measurement. And this makes your cluster activity more robust to noise, okay? In other words, we are going to have more robust predictors. And also clustering help us to reduce the collinearity among our predictors. So it serves multiple purposes. And it turns out that using the clusters as the predictors rather than using individual genes as predictors, you actually can get better prediction performance. Therefore, our predictors are built upon the clusters. And after we compute the cluster level activities, we apply a fast variable screening procedure to choose the clusters that are most informative for each individual genomic locus. For example, for the first locus, we are going to use a variable screening procedure to choose a subset of clusters that are most predictive for that locus. And then using those selected clusters, we're going to build a regression model and as our prediction model for that locus. And we are going to repeat this exercise for each and every genomic locus. And that will give you a locus level prediction. And since we clustered genes in the gene expression matrix, a natural question is, why don't you also cluster cis-regulatory elements? Actually, we did that. So we also clustered the co-activated cis-regulatory elements into clusters. And for each cluster, again, we can use their average patterns to summarize the cluster pattern. And we repeat a similar exercise. We choose some of the predictors that are most informative for predicting cluster level activities, and we then trained prediction models to predict the cluster level activities for each co-activated regulatory element clusters. And then our final prediction for the cis-regulatory activities combines the locus level prediction and the cluster level prediction through a model aggregation procedure. Why do we want to combine both? So the intuition is, if you train a prediction model for each individual locus, you can get an unbiased prediction in some sense. However, the measurement for each locus is quite noisy. Therefore, your prediction model will have high level of noise. Okay? When you cluster co-activated cis-regulatory elements together and compute the mean activity, you reduce the noise such that the cluster level activity can be more precisely accurate in the sense that the variance of your prediction will be smaller. Okay, and then if you use the cluster level activity to represent the locus level activity, obviously you are going to have some bias in terms of describing the locus level activity. However, since the cluster level activity can be predicted with smaller variance, it gives you a leverage to somehow balance the variance and the bias. If you balance the bias and the variance well, it actually will generate better prediction for each individual locus. And for that reason, we construct our prediction model both for each individual locus and for cis-regulatory clusters. And then we combine them together using a model averaging strategy. And the details of how we combine the model actually can be found in our published paper. So I will ignore those details, but the intuition is by combining these two, you actually improve the prediction accuracy. So that's our bird approach. So in this approach, essentially, we are using the whole transcriptome information to predict the genome-wide cis-regulatory element activities. So how does this approach work? We evaluated this approach using using the ENCODE data we have collected. And as I mentioned, we have collected 57 distinct human cell lines with both DNA-seq and exon-ray data. And we have partitioned these 57 cell lines into a training set consisting of 40 different cell types and a test set consisting of 17 different cell types. And we use the training data set to train the bird model we have, and then applied the trend model to the 17 test cell types to see how we can predict. And on the bottom, you can see 5 different predictions in 5 different cell types. And on the left-hand side are the true DNA-seq data. So these are the true activity levels measured by experimental DNA-seq. And on the right-hand side are the BERT predictions. And each row corresponds to a cell type. And this is predictions in a sample genomic region. You can see that the BERT predicted cis regulatory element activities captured the true cis element activities reasonably well, and the main variation both across locus and across cell types are captured by our prediction. And we actually did a more systematic comparison, more systematic evaluation of our prediction performance. And for the testing cell types, we made predictions, and you can organize the predicted value again into a matrix. Each row corresponds to a cis element, and each column corresponds to a test cell type. And since these are from ENCODE, we actually have the true DNA-seq data. We use that as the gold standard. And again, each row is a locus, each column is a cell type. And then we computed different evaluation statistics. And the first measure we used is the so-called cross-locus correlation. And to compute cross-locus correlation, what we did is for each cell type, for each column of these matrices, we compute the correlation between the predicted value and the true value across different genomic loci. So that's the cross-locus correlation. And you can see that the cross-locus correlation of BERT can be is around 0.8, so which is pretty good. And we actually also compared the prediction performance from neighboring genes. Remember, we actually first tried the neighboring gene approach, and in which we selected neighboring genes of each cis regulatory element and constructed prediction models. And the prediction performance using the neighboring gene approach actually was worse than using the BERT to do the prediction. And we also compared it with some random permutation models. And again, BERT significantly outperforms the random prediction models. And the second measure we use to evaluate our performance is the so-called cross-cell type correlation. In other words, for each genomic locus, for each cis regulatory elements, we computed the correlation between our predicted values and the true values across different cell types. Okay, and again, you can see that the BERT prediction significantly outperformed the neighboring gene approach, and also significantly outperformed the random prediction models. And finally, we have evaluated our prediction using the squared prediction error. That is, we compute the difference between our predicted value and the true value for each element and each cell. And then we take square of that difference and somehow normalize by the total data variance. And the smaller the error, the better. And you can see, again, BERT offered the smallest squared prediction error compared to the neighboring gene approach and compared to the random prediction models. So that means in the overall evaluation, BERT actually outperformed the other methods. And if you carefully look at the prediction performance, you can see that actually the random models did a pretty good job in terms of predicting cross-locus variation. The cross-locus correlation for random prediction models actually has a mean of 0.65. That's very high, right? So initially we were surprised by this, but when you carefully look at the data, it sort of makes sense. This is because this slide shows the DNA-seq data for many different cell types in a sample genomic region. You can see that certain genomic loci have much higher activity levels compared to other genomic loci, like the loci here, regardless of the cell type. And for that reason, if you use the mean DNA-seq profile of all cell types as a predictor, you actually can predict the DNA-seq profile in a new cell type reasonably well. That's why the random prediction model actually did a reasonably good job for predict cross-locus correlation. However, the mean profile of all these cell types will not change based on like new cell type. Therefore, it cannot predict the cross-cell type correlation very well, right? So that's why when you look at the cross-cell type correlation performance of random prediction models, it actually performed very badly, and the correlation is about 0. Okay, so that's a small lesson we have learned. So anyway, so we have developed these BIRD methods, and it can predict both cross-locus correlation and cross-cell type correlation. And when you compare these two correlations, you can see that cross-cell type correlation usually is lower than cross-locus correlation because of the locus effect I have just shown there. However, cross-cell type prediction accuracy can actually increase if you focus on studying like loci that show high variability. And we actually did some further analysis in which we have shown that you have loci that's highly variable across different cell types, you actually can increase the prediction accuracy to 0.6 or 0.7. Okay. And we have compared BIRD with many other methods such as k-nearest neighbor, random forest, lasso, and stepwise regression, and so on and so forth. And in all these comparisons, BIRD actually offered one of the best prediction accuracy, actually is the best prediction accuracy. And also it is computationally very efficient. And for example, compared to random forest, our BIRD is more accurate, and its running time is orders of magnitude lower than random forest. And for whole genome model training and testing, our BIRD will take around 1 or 2 days, depending on what type of computer you have. But for random forest, our test based on 1% of genome give an estimate of like you may need 1 month or even more than that to finish a whole genome run. Okay, so for that reason, we think that BIRD is very attractive in terms of both computational efficiency and the prediction accuracy. And we have also compared BIRD with another method called chromimpute, and many of you perhaps know chromimpute. And this is a method people develop for imputing one epigenomic data type using multiple other epigenomic data types. And we applied chromimpute to impute DNA-seq using multiple histone modification marks, as well as DNA methylation and many other epigenome signals. And surprisingly, BIRD and chromimpute performed comparable. So we have tested both algorithms in 10 different cell types, and BIRD outperformed. BIRD is the green one, chromimpute is the blue one. And BIRD outperformed chromimpute in 5 of these tests, and chromimpute outperformed BIRD in the other 5 of the test cases. And that suggests that both algorithms have a similar prediction accuracy. However, remember that the chromimpute uses many histone modifications as its predictors, and BIRD only uses gene expression. So that's a huge difference, because histone modifications usually their locations coincide with the open chromatin regions, so they actually intuitively they should be more informative. And also chromimpute use multiple histone modification as the predictors. So before we do this comparison, I thought it was like chromimpute probably should perform better than BIRD, but actually it was not the case. And it's somehow surprising that using only using only the gene expression data, we can achieve a similar prediction performance compared to chromimpute. In terms of practical utility, we know that the number of gene expression samples in the database is much larger than the number of histone modification or other epigenomic data types. Therefore, BIRD arguably is more widely applicable, because you only need gene expression data to do this prediction, whereas in chromimpute, you would need collect many histone modification data types, and those experiments themselves are not easy to conduct. Okay, and then we ask whether we can use the BIRD predicted DNA's hypersensitivity to predict transcript factor binding sites. And to do that, we mapped, for example, here I used a prediction of transcription factor MYC binding sites as an example. To predict MYC binding sites in a B-cell lymphoma cell line, we mapped a MYC motif site to the genome, and then we applied BERT to an exon array sample for that cell type, and then we have predicted DNA-seq. Combined with the DNA motif information, we can then predict the MYC binding sites. And comparing to the MYC chip-seq, so chip-seq is the current state of the art to map the binding sites, right? We used it as gold standard. Compared to the MYC chip-seq, you can see that our BERT prediction actually can accurately predict MYC binding sites. For example, when you look at the bottom left figure here, the red track are the BERT predicted binding sites, and the blue track represents the MYC chip-seq, which is the gold standard. And our prediction captured the gold standard very well, whereas if you just use the mean DNA-seq profile of all cell types, it does not predict the MYC binding activity. So this shows actually BERT can predict, combined with DNA motif information, BERT can predict transcription factor binding sites. And then we performed a more systematic evaluation in which we have predicted binding sites of many different transcription factors. For each transcription factor, we have generated this sensitivity and force discovery rate curves. And you can see that BERT actually performed very well. So take the ELF as an example. And the blue curve shows the sensitivity versus force discovery rate for predicting transcription factor binding sites for ELF1 in one cell line, GM12878, and that's the curve. And then as a positive control, we used the experimental DNA-seq data combined with DNA motif information to predict binding sites. And intuitively, that experimental DNA-seq data should perform better. But you can see like BERT actually performed comparable to predictions you would obtain if you have the real experimental DNA-seq data. Okay? And then compared with the motif-only method, if you only predict the binding sites using DNA motif without using DNA-seq information, that is what you are going to have. And you can see that BERT significantly outperforms the motif-only approach. And another approach we compare is the mean DNA-seq profile. If you only use the average DNA-seq profile across all training cell types as the predicted DNAs and combined with DNA motif information, the red curve is what you would obtain. And again, BERT performed significantly better than that approach. So we did this experiment in many other cell types and for many other transcription factors. The message is similar. That is, BERT actually outperformed the motif-only and the mean DNA-seq approach, and sometimes can perform comparable to using true DNA-seq data to predict transcription factor binding sites. Okay. And then we have actually applied BERT to 2,000 public available exon array samples in the Gene Expression Omnibus database. And we have created a web database resource called PDDB, the Predicted DNAs database, for users to use. And if you are interested, you can search the database by cell type, by genomic region, and so on and so forth to get the predicted activities of the cis-regulated elements you may want to study. And this database can be used, for example, to help you interpret GWAS SNPs or to help you predict which functional element you may want to perturb in your functional studies. Okay, so this is a resource. All right, so up to this point, what I have talked about is using exon array data to do the prediction. So after we have done that pilot study, the next question we ask is whether we can use RNA-seq to do the prediction, because RNA-seq is now state of the art of transcriptome profiling. And to do that, we repeated our exercise. We collected the cell types for which people have collected both DNA-seq and RNA-seq data from the Roadmap Epigenomics project. And then we trained the bird models using the approach I described above. And then using that prediction model, we then tested the prediction performance in independent samples measured by bulk RNA-seq. And on the right-hand side, you can see this is an example genomic region. Again, each row corresponds to a cell type. I have shown 5 different cell types here. And the bottom plot shows the predicted DNase1 hypersensitivity in that genomic region. And the top plot shows the actual DNase-seq data. And again, you can see we predicted the true DNase reasonably well. And we again did the genome-wide evaluation, and the message is about the same. That is, we can predict the DNA-seq very well using the genome-wide RNA-seq data. And then we asked whether we can predict using RNA-seq samples with small number of cells, because we know that in precious clinical samples or when you study early embryos, sometimes you don't have that many cells. And therefore, it is very important that you can do prediction in those scenarios. And surprisingly, what we found is that bird prediction using samples with small number of cells sometimes can perform better than bulk ATAC-seq, which is the current state of the art for chromatin accessibility profiling in small cell number samples. And here, what I'm showing you is one example. And here we want to predict chromatin accessibility in a test cell type GM12878. And we have applied bird to a bulk RNA-seq sample obtained using 10 cells. And we have applied bird to a RNA-seq sample generated using 30 cells. And we have also applied bird to a sample generated using 100 cells. Okay, and then this bar, this number here, shows the correlation between our prediction and the true bulk DNA-seq, which is viewed as the gold standard. And the signal here shows the predicted values in a sample genomic region. Okay, as gold standard, you can see in the bottom track here, we have shown the true DNA-seq. These are bulk DNA-seq data, and you can use this as the gold standard. And you can see that bird prediction using 10 cells, 30 cells, 100 cells all provided reasonable performance. And interestingly, when you look at this track is the experimental ATAC-seq data generated using 500 cells. The correlation between that ATAC-seq and the bulk DNA-seq is only 0.63, which is comparable to our bird prediction using 10 cells. If you look at the bird prediction using 30 cells, it actually outperforms ATAC-seq using 500 cells, okay? And that says we can use a smaller number of cells to achieve a better accuracy in terms of measuring chromatin accessibility. And when you use ATAC-seq on 50,000 cells, it actually performed better than bird. However, if you combine our bird prediction, so using, for example, using a RNA-seq sample generating 30 cells, we can make a bird prediction, right? And we can treat that bird prediction as a pseudo-replica sample and combine it with a experimental ATAC-seq sample generated using 500 cells. And we combine it by computing the average between our bird prediction using 30 cells and the ATAC-seq data obtained using 500 cells. So in total, we used 530 cells. And if you do this, you can see the prediction accuracy actually is as good as the ATAC-seq generated using 50,000 cells. Or in other words, using only 530 cells, we actually achieve the same accuracy as experimental ATAC-seq data, which is obtained using 50,000 cells, okay? And then we actually can also improve ATAC-seq using 50,000 cells. And again, here what we did here is we use bird prediction as a pseudo-replicate sample. In other words, we first predict using an RNA-seq sample with 30 cells, And then we use the predicted values as pseudo-replicate and combine it with ataxic generated using 50,000 cells. And we compute their average. And you can see this average correlated much better with the DNA-seq sample compared to the ataxic itself. And the correlation with the gold standard increased from 0.76 to 0.8. In other words, using about the same number of cells, eventually we performed better than ataxic. So a message learned from this exercise is that BERT actually can predict chromatin accessibility very accurately in samples with small number of cells. So when you don't have DNA-seq or ataxic sample, when you only have RNA-seq sample, you can make this prediction to help you obtain that information. And even if you have the ataxic or DNA-seq sample for those in those situations, you can still use BERT, because you can always use BERT prediction as a pseudo-replicate and combine it with experimental DNA-seq or ataxic data to further improve the signal quality. So that's the lesson learned from this exercise. So this plot shows some example correlation. For example, we have compared bulk ataxic with bulk DNA-seq, and the correlation is 0.76. And when you compare bulk ataxic from 500 cells, the correlation is lower. However, if you use BERT prediction using 30 cells, the correlation with the gold standard bulk DNA-seq actually is higher compared to the ATAC obtained from 500 cells. And if you combine BERT prediction using 30 cells and experimental ataxic data using 500 cells, it actually achieved the same performance as ataxic using 50,000 cells. So that's an example to illustrate the power of BERT. And next, we ask whether we can apply BERT to single-cell RNA-seq data. So why are we interested in single-cell RNA-seq? Because this is a single-cell technology, it's a powerful technology that allows you to study the behavior of each individual cells. And it allows us to identify real cell populations and characterize their properties. And many of these things cannot be done using the traditional bulk technology. And currently, actually, there are technologies to measure chromatin accessibility at the single-cell level. So the widely used method is the single-cell ataxic. However, single-cell ataxic has its own limitations. And one limitation for this technology is that it is highly sparse and the data is almost binary. And when you look at the single-cell ataxic data, you can see that at each genomic locus, usually you only have 0 read or 1 read or 2 reads. So that's it. Why is that the case? Because single-cell ataxic tries to measure chromatin, and within a single cell, there are only 2 copies of chromatin at each locus. And therefore, the number of non-redundant reads you can obtain is at most 2 in theory. Right? However, we can argue that the cis-regulatory element activities is not a binary signal. It actually is a continuous signal. This is because transcription factor binding and dissociation event. So the binding of transcription factor to DNA and dissociation of transcription factor from the DNA is a stochastic event. It's a temporal stochastic process, actually. At one time point, the transcription factors sit here. Next time point, the transcription factor may dissociate from DNA. And then after a while, the transcription factor binding may occur again. So this is a temporal stochastic process. Therefore, the activity of each cis-regulated element actually should be described by the probability that the transcription factor will sit there at a random time point. And the probability is a continuous measure between 0 and 1, rather than a binary measure 0 or 1. Right? So therefore, the sparse and binary single-cell ataxic data cannot accurately capture the activity level, steady-state activity level of each cis-regulated element in one single cell. So that's the intrinsic limitation of single-cell ataxic. So we wondered whether we can use single-cell RNA-seq to better predict chromatin accessibility. So in the single-cell RNA-seq, each gene can have multiple transcripts. Therefore, the single-cell RNA-seq data, in principle, can be more continuous, right? So we we want to ask whether we actually can leverage that continuity to do a better job. So we trained our BIRD model using bulk DNA-seq and bulk RNA-seq data from the ENCODE. And then we applied those trained model to several single-cell data sets. And surprisingly, our predictions based on single-cell RNA-seq performed better than single-cell ATAC-seq. So this plot shows one example in one genomic, example genomic region. Again, this is a prediction in a well-studied cell line, GM12878. And the bottom track shows the bulk DNA-seq, and the top track shows the bulk ATAC-seq. And you can use the top track and the bottom track as the gold standard. So the three red tracks shows the BIRD prediction using single-cell RNA-seq. Here, what we did is we make prediction. We put 10 cells of the single-cell RNA-seq together and made prediction. And these are the predicted value. You can see the predicted value correlated with the gold standard very well. And if you put 28 cells, again, you correlate with the bulk DNA very well. And the green and the purple tracks are experimental single-cell ATAC-seq data generated by two different labs. Okay, and then here we did the same thing. We put 10 cells or 28 cells together, and you can see that given the same number of cells, BIRD prediction actually better captures the DNA-seq or bulk ATAC-seq pattern compared to the single-cell ATAC-seq. Okay, so this is only one example. We did a more systematic evaluation. So in this evaluation, we used the BIRD to predict using single-cell RNA-seq, and we also compared that prediction with the single-cell ATAC-seq data from two different labs. And for each approach, we put different number of cells ranging from 1 cell to 5 cell, 10 cell, 20 cell, 28 cell, and so on and so forth. And with the same number of cells, we compared the accuracy of different methods. And you can see the red curve, which represents the accuracy of the BIRD, which is the correlation between the BIRD predicted value and the true DNA value, consistently. So the BIRD performance consistently outperformed the single-cell ATAC-seq in terms of reconstructing the bulk chromatin accessibility. So this shows that actually BIRD can use single-cell RNA-seq to better predict chromatin accessibility compared to the state-of-the-art single-cell ATAC-seq technology. And we did the same analysis in different cell types. For example, this is another cell type, H1 embryonic stem cells. The message is the same. We again outperformed the single-cell ATAC-seq. And we also combined our predicted values with DNA motif information to predict transcription factor binding sites. Again, the message is the same. With the same number of cells, transcription factor binding sites predicted using BIRD actually outperforms the transcription factor binding sites predicted using experimental single-cell ATAC-seq data. So I will skip the detail here. And we also have predicted differential regulatory activities between two different cell types, say between H1 and the GM. And we can predict the differential activity of each cis-regulated element. Again, the BIRD prediction, the red curve outperformed the experimental single-cell ATAC-seq in terms of capturing differential cis-regulated element activities. Okay, so these are just examples that I probably would skip. And finally, I'm going to discuss a real example. So in this real example, we tried to analyze single-cell RNA-seq data generated by the Human Cell Atlas project for studying the hematopoietic differentiation. And this differentiation is a complex process where the hematopoietic stem cells differentiate into different lineages. So the Human Cell Atlas actually collected the bone marrow data from 8 different patients. And here, what I'm showing you is data for 1 patient. And for that patient, we first visualize the single-cell RNA-seq data, and you can see there are different cell types, and the blue in the middle represents the embryonic, the human hematopoietic stem cells, and the green, red, and the purple represent 3 different lineages of differentiation. Okay, and some of them are like lymphoid lineage, some are like monocyte, and the others are the red blood cells. So, so essentially they represent 3 different lineages. And then we have used the pseudo-time construction method developed by ourselves to construct a pseudo-time, which captures the developmental trajectory of these cells. So this pseudo-time is constructed using single-cell RNA-seq. And then after we construct this pseudo-time, along each pseudo-time, we then apply the BERT to predict, for each cell, we can predict what is its gene regulatory landscape, right? So that's what we have obtained. And we know that there are 3 developmental lineage, differentiation lineages, which are represented using the 3 columns here. And for each lineage, we have visualized the BERT prediction using a heat map. Here, each row corresponds to a cis-regulatory element, and each column corresponds to a time point in the pseudo-temporal trajectory. Or you can think it as a cell, okay? So each column can be thought of as a cell, and each row is a cis element. And you can see that we have identified a bunch of cis-regulatory elements that decreases along this differentiation lineage, and we have identified another set of cis elements that increases the activity along this lineage. And for these other 2 lineages, we also identified different sets of cis-regulatory elements that either decrease or increase their activity. And to see whether our prediction is correct or not, we actually analyzed a separate set of single-cell ataxic data for the same system. And you can see that for the same, so in these 2 plots, so the bottom plot are the results we obtained from experimental ataxic data. And each column here corresponds to the same column in the plot above. They represent the same cis-regulatory element. And you can see that our prediction actually captured the pattern you would obtain from the experimental ataxic data very well. So that means actually we can use our prediction to study the gene regulatory dynamics in a single-cell RNA-seq data set. And here, by the way, the single-cell RNA-seq is generated using 10x genomics. So it shows that in a 10x genomic data set, if you don't have ataxic or other regulum data, you actually can use BERT to collect the information on cis-regulatory element activities. And you can actually use this to study the dynamic changes of gene regulatory landscape along a dynamic process, for example, along the pseudo-temporal trajectory. Okay, so this is an example. With this example, I'm going to conclude my talk. So to summary, I have shown that regulum or the genome-wide regulatory element activities actually can be predicted using gene expression. And we have developed BERT, big data regression and prediction, to solve this problem. And it also shows that public data is a powerful tool to make discoveries, because when you think about BERT, what we did is just we used massive amounts of public data in ENCODE or in epigenome roadmap data to train prediction models, and then use those prediction models to help us gain more information from the newly generated bulk or single-cell RNA-seq data. And prediction can actually be used to map regulum in massive number of biological contexts. We have shown that by applying BERT to the samples in the gene expression omnibus. And one ongoing project that we're pursuing is we are trying to improve our BERT prediction so that we can make predictions for most of the gene expression samples in the gene expression omnibus. That will convert GEO into a regulum database if we can do that. And finally, when there is no single-cell ataxic data, my final example shows that using BERT, you can actually predict gene regulatory dynamics using single-cell RNA-seq. And in terms of future directions, there are plenty room to improve our prediction methods. For example, for the single-cell prediction, single-cell RNA-seq data actually has a lot of technical biases and noises, and we are working on improving methods for handling those biases and noises, and hopefully that can help us further improve the single-cell prediction accuracy. And also in terms of training prediction models, currently our training data needs to be matched in terms of DNA-seq and RNA-seq. And we are exploring semi-supervised learning methods so that we can use unmatched training samples, meaning that cell types with only one data type, say RNA-seq or DNA-seq, but not both. We want to use those unmatched training samples to further improve our prediction model training. So that's another direction we are trying to explore. And finally, multi-omic single-cell analysis, and we are trying to explore methods to use BERT prediction as a bridge to help us integrate different data types. And currently, we are exploring ways to integrate single-cell RNA-seq and single-cell ATAC-seq through BERT predictions. So there are also many other possible future directions. But anyway, so this is a very exciting new area, and I think a lot of work can be done in this area. And finally, if you are interested in the details of the work I presented today, these are two references. And the first one is already published, and the second one is currently under revision, but you can find a preprint in bioRxiv. And I would like to thank the members in my group who participated in this work, and including Wei Qiang, Ben, Zhicheng, Fang, Jiawei, and Wei Xiang. And I also want to thank funding agencies for providing fundings for this research. And I'm going to conclude my talk now, and thank you for your attention.
All right, thank you, Dr. Ji. That was a fantastic talk. So we still have about 7 to 8 minutes to wrap up, and everybody's still muted, but if you have any questions, you can unmute yourself with questions and kinds of comments. So I'll just be the first to get started. So Dr. Ji, that was a really, really good talk. I have about 2 questions. One is really focusing on the high level, and the other is sort of technical. So could you make a few comments on, because my understanding of how BERT works is that it will not, I don't think it will be able to discover, for example, new locations of DHS when we have new sample coming in with gene expression profiled. So could you make a few comments on how can we sort of resolve that, in identifying or identification de novo or new locations of DHS as predictions in your methodologies?
That's a great question. That's the question we got from almost all reviewers. So, and to answer this question, we actually have done a saturation analysis, and we have collected all the encoded DNA-seq data, right? So, and then we can do a saturation analysis and ask how many new regulatory elements you can find in a new sample, right? Mm-hmm. So it turns out that in that saturation analysis, what we observe is that in any new sample or new data set, for example, new single-cell ATAC-seq data set, the number of new cis elements you can find usually is less than 0.1% compared to the cis regulatory element we have already compiled using the public data. So that means most of the vast majority of the cis regulatory elements are already covered by existing data. What we don't know are their cell type specific activities. We actually know their locations, but we don't know their cell type specific activities. So that's where BERT actually helps, right? And in terms of discover the new cis regulatory elements, and in certain scenarios, you can still do that. For example, if you have some single cell data, single cell ATAC-seq data, you could sort of first use some mediocre quality single cell ATAC or bulk ATAC-seq data to get a new element. And then if you have RNA-seq data, you can still do your prediction on those new elements, and then combine the prediction with the experimental measurement to further improve your quality. So in both of the cases, BERT are still useful.
I see. Okay, thanks. And my second pretty technical question is, I don't know, through your public data search, when you're training your models, have you ever looked at or compared the performance difference between different RNA methodologies. And specifically, what I'm asking is, I don't know if you have ever compared performance in total RNA-seq data, where you also not only have the exome expression, you will have non-coding regions, compared to, for example, Illumina has this kit, it's called TrueSeq Exome Kit, that is only profiling the whole exomes. Usually you get deeper coverage of the transcriptomics. I just don't, because my understanding is for BERT to work better, you need to accurately profile the motif information. And I don't know, like different RNA-seq platforms, if you see like some discrepancies or differences of the performance.
Again, that's another great question. We haven't specifically done that comparison you suggested. However, we did apply BERT to both bulk and single-cell RNA-seq, right? So if you think these as two different platforms, we actually have some empirical evidence from our exercises here that suggests that regardless of the platform, BERT actually can generate reasonably good predictions, right?
Okay, cool. Anybody else, if you have questions, you can unmute yourself and ask. All right. I do have one more technical question, Dr. G, if you don't mind. So in order for us to run this BERT algorithm, I think it's probably available online somewhere on GitHub, but can you make a few comments how computationally expensive this approach is? For example, we have like 500 or maybe 100 new samples coming in and we want to infer.
Yeah. So it depends on what you want to do. So we have in our published paper, you can find the GitHub link to the BERT software. And if you only want to use the trend model to make predictions in new samples, for example, if you have some new human RNA-seq samples, you want to make prediction on them, you actually don't need to retrain the model because we have included the trend model in the website. So you can directly download those trained model, and making prediction is quite fast. Like within a couple of seconds, you can handle hundreds of samples. Okay, so that's easy. On the other hand, if you want to train the model yourself, then you probably would need a longer time. And if you have all the data organized, we actually also uploaded the training data to our website. If you just want to use our training data, that will save your effort. And then say you have some new RNA-seq and DNA-seq samples, you want to add them to the training data to expand it, then you need to do some kind of normalization for which we have provided functions. And then that may take, say, one day if you are like familiar with all those R packages, right? And then once you have the training data ready, training the model usually only take 1 or 2 days, the genomewide model. So once you train the model and the application again is only a couple of seconds to the new samples.
I see. All right, I think we are right on time. I want to thank Dr. G for coming to the Journal Club and giving this fantastic talk. And thank everybody for your attention. If you have further additional questions, feel free to contact us or just email us or message us through WeChat, and we would definitely point you to the right person to answer that questions. Stay tuned for our next coming event of DaShu. And again, thanks everybody for attending this event. I wish you have a very good weekend. Thank you, everybody. Thank you, Dr. G.
Thank you.
Okay, bye-bye.
Bye.